Hi, everyone. Welcome to our Tuesday seminars on the cerebellum. Today we have as our distinguished guest, Abby Person. Abby did her PhD at the University of Washington, working on the songbird system, discovering some of the anatomy of the basal ganglia and some of the learning rules of the thalamus. Then she did a short stint in Beijing, followed by a postdoc at Indira Raman's lab at Northwestern University. And then she got a position at the University of Colorado, where she has been doing fabulous work, basically giving us so much knowledge about the relationship between Purkinje cells, their firing activity as a population, the notions of synchrony and control of movement. She is a Sloan Fellow and a Mcknight Scholar, and she is an amateur archaeologist, and makes her one of the best persons that I can think of to take a walk in nature, because she's very much like a forest person that can guide you. So with that, Abby, thank you for being with us.
Reza, thank you so much. So I'm going to put on my forest person cap, and we're going to go through this journey of nature together. I think that I was just talking with a student the other day about what an immense privilege it is to be a scientist and to have this opportunity to explore nature in ways that are, at the forefront of knowledge. And so it's really a wonderful joy to have this profession and to have you all as colleagues, because it makes it wonderful to have a chance to share ideas and learn from you. And as you'll see, Reza has inspired numerous concepts that we've explored in here. So that's my preamble. So I'm going to start with this. I may start with this figure that's taken from a study by Gordon Holmes, who was a British physician during the time of the First World War. And he, like many physicians, would perform standard neurological exams on veterans who had been injured at the front. And he noted that there were many of these patients coming to him with very severe movement disorders, and they seemed associated with gunshot wounds to the back of the head, which was a part of the head that was not well protected with the style of helmets that was used at the time. And so he had this idea that he was going to track these movements and be able to record them. So Reza, here we have your, sort of version of the clay tablets. Like, this is a recording of the movement that you otherwise would not have any record of. And he used this clever method by attaching a light bulb to the finger of the patient. And then he used long photographic exposures during this neurological exam. And so the common neurological exam that's used to this day in neurology clinics is to have the patient point from their nose to 3 different points in space, just following the finger of the physician. And so what you can see from this trace is that during these movements, there were pronounced oscillations near the end points of the pointing movements. And these oscillations, you can see them and they characterize many forms of cerebellar damage. And so I'm going to show you what this looks like in this man who has a unilateral cerebellar infarct. He's going to point to his nose with his good hand, and then he's going to point to his nose with the hand that's ipsilateral to the cerebellar damage. So here's the good side, and then he will point with his affected hand. So you see that very prominent oscillation appear. And this is what Gordon Holmes also had observed. So this means that these endpoint oscillations are a characteristic hallmark of cerebellar damage. And what's interesting about them per se is that they resemble the kinds of oscillations that commonly occur in feedback-driven control systems, such as thermostats or cruise control systems that are poorly tuned. Okay, and so the basic idea is that anytime you have a control system that uses feedback with a considerable delay relative to the output, oscillations tend to emerge if that feedback is not well damped. Okay, so because the body produces endpoint oscillations without the cerebellum and does not produce those oscillations with the cerebellum, it suggests that the cerebellum is performing one of these control system solutions. And so that's the general framework of ideas that we explored as we began the lab. And the overarching framework is that the cerebellum may compute some sort of feed-forward control signal that precludes the formation of these kinds of endpoint oscillations. So with this as a basic conceptual framework, we could then ask, where within the circuit might we be able to observe these feed-forward control signals, and how might they be learned? So here I have a sort of box diagram that is mapped onto roughly the cerebellar circuit over here. And so I want to walk through the flavors of information that come into the cerebellum, how they may be transformed, and where we would go looking for a feed-forward control signal if it actually existed. So the general thought that's emerged through decades of work by cerebellologists is that the major input to the cerebellum through the mossy fiber systems carry a mixture of sensory feedback or sensory information from the periphery, as well as motor control signals that go by the term corollary discharge or efference copy signals. And both of those streams of information that enter through the mossy fiber systems get carried into the granule cell layer. The granule cells you can think of like a prism that take all of this sort of mixture of white light and break it into its component colors. It separates information so that you can see it more deeply. And by you, I mean, if you're a Purkinje cell, you can see the sort of depth of that information that is produced by the granule cell layer. So that sort of represents the context of the system. In this case, this is usually the context of movement, but it could be context of all different sorts. So that's then projected to the Purkinje neurons, which of course are the most magnificent cells in the brain. They're very famous for their beauty, and they're also famous for their plasticity. They are the magnificent learners of the circuit, and they then are inhibitory neurons also and project into the nether region of the cerebellum, which is nevertheless the heart of the cerebellum, which is the cerebellar nuclei. So those are the output structures. The interposed nucleus is one of them, of course. And so if we wanted to say that, the cerebellum may compute a feed-forward control signal, this is where you would go and look for it, because this is the output of the cerebellum, right? And yet the cerebellar nuclei have received relatively little attention for a variety of reasons. Most of these reasons are technical. It's hard to get to them. Their signals are diverse, and so it's been hard to study them. But nevertheless, we have sort of started our investigations by starting at the end of the cerebellum and working backwards. So as I've kind of alluded to, if we want to go in search of a feed-forward control signal, where do we look and what do we test? Well, it turns out that there was a whole body of research that had been developed over decades that suggested that the anterior interposed nucleus is involved in limb control. So this is a study from Claude Getz's lab, where they injected a reversible inactivator, a GABA-A agonist, muscimol, into the anterior interposed nucleus in cats that were performing a reach-to-grasp task to retrieve a small piece of meat inside of a box. And these are the wrist paths, and they observed that in general, these reaching movements were hypometric under these conditions. Later on, a beautiful study from Albert Chen's group ablated neurons in the anterior interposed nucleus in mice that were performing a reaching task. And in this instance, they observed that mouse reaches were hypermetric. So they were going too far. Now, these studies are really important for a variety of reasons. They tell us that the anterior interposed nucleus may be involved in structuring reaches, and that the endpoints are, maladaptive in some way. But one of the challenges of interpreting these longer-term inactivation or lesion studies is that they don't distinguish active control principles from movement initiation, movement planning, and they don't identify a control policy. So those are really the questions that are perfectly suited to electrophysiology. And they motivated the experiments that were done by my student, Matt Becker, when he was in the lab. He's now at the University of Washington, and he's doing a psychiatry residency with a research emphasis. And so we're excited to see what Matt does next. But anyway, when he was in the lab, he used tetrad recordings targeted to the anterior interposed nucleus in mice, and then had them perform this forelimb reaching task that was developed by Ian Wishaw years ago. And so he also used high-speed kinematic tracking so that he could very well relate neural activity to the behavior. So here's what a reach looks like in a mouse that's reaching to grasp a food pellet. And Matt observed that there were many neurons in the anterior interposed nucleus that had neural activity that was seemingly tuned to the endpoint of reaches. This was a relatively common observation in that there were many of these neurons that had their peak activity during this decelerative phase of the reaching movement. And so the natural question was, does this neural activity have anything to do with the reach at all? Is this reporting something, or is this causing something, or both? And so to test that, Matt combined this real-time kinematic tracking with optogenetic manipulation of neurons in the anterior interposed nucleus. And so basically the idea is that since we have real-time positional information, he could trigger optogenetic perturbations of these neurons in closed loop with the reaching movement. And he did this on a randomized 25% of trials to prevent learning. And what he observed was something like this. So here the mouse is receiving a very brief 50 millisecond train of pulses during this period. And you can see that the mouse does not get the pellet. Okay, so sometimes the mouse doesn't get the pellet, so let's look at lots of reaches. So this is a plot that shows many reaches. The unstimulated reaches are shown in orange, the stimulated reaches are shown in blue. And if we pull out a still of this just after the stimulation time, which is shown here, we see that the stimulated reaches are not as far out and they're not as far up. So they end in this hypometric position. And so this is interesting, given the fact that there's all this neural activity near endpoint. Is this causing the limb to slow down? So Matt did a series of other experiments where he modulated the amplitude of the laser, and there was a scaling of the deceleration. And he also could stimulate at any point in the reach and cause this slowdown. And so that collection of data suggested that the anterior interposed nucleus may dictate when and how fast the limb should decelerate. Now, if we're right about that, we should be able to do the opposite experiment, take spikes away and see an opposite kinematic effect. So the next experiments Matt did was to introduce an inhibitory opsin, ARCH, into the anterior interposed nucleus, and again, inactivate these neurons in closed loop with reach. And indeed, what he saw in all animals was an opposite kinematic effect. That is, the limb remained faster longer, and reaches ended up hypermetric. So they went too far. Now we're a little bit at sort of a biomechanical barrier here because the reaches are very near the sort of maximum extent of the limb. But the upshot of this whole series of experiments is that activating the anterior interposed nucleus caused a reduction in outward velocity, and inactivating it caused a increase in outward velocity. Now, there's many ways to interpret what is going on in this situation. And so, two possible models is that this neural activity in anterior interpose could be acting as a brake or a gain controller. In here, I'm talking about a brake as in like a brake in your car. Okay, and so that's consistent with the data that we observed, right? We have, let's say, a hypothesized control reach that doesn't have this brake, and when we stimulate, it doesn't go out as far. But the data are also equally consistent with an alternative hypothesis, which is that maybe there is a directional command that's being issued by the anterior interposed nucleus. And we could think of that command as having a directional vector like this. And if it were added onto this hypothesized control vector, you would also see the same thing, what we saw. Now, what's interesting about these two hypotheses is that they make distinguishable predictions about what should happen if we stimulate on return. If there's a directional command, we would expect actually that the limb would move back faster. However, if it's just a brake, then we would expect the limb to move back slower if we stimulate on return. And so we performed this experiment, and what we observed is consistent with this directional command hypothesis. So here we are stimulating in the negative velocity space, so when it's returning back, and the limb moves back faster. Okay, so this is, like I said, suggests that there's a summation of a directional cerebellar command with ongoing downstream control signals that are likely issued from other regions like motor cortex. And so with this body of information in hand, we went back to the electrophysiology and said, can we identify any kind of a feed-forward limb control sort of mechanism or principle that's at work? So I showed you this, this raster plot before, and the first prediction from the optogenetic data is that we should see a relationship between the firing rate on a given trial and the rate of deceleration of the limb. So in some simple linear regressions of neurons that were active during this decelerative phase, we found consistent relationships between the firing rate and the magnitude of deceleration. So great, this all makes sense. But it raises the question of why might there be trial-to-trial variability of deceleration and the concomitant firing rate. So Matt did this analysis of this data where he pooled reaches based on their firing rates into deciles. So the reaches that had the very highest firing rate are shown in red, and those with the lowest peak firing rate are shown in blue. And consistent with the linear regressions, the reaches with the lowest firing rates decelerated more slowly than those with the highest firing rates. But again, the question is, why might that be? So then he went back in time, so before these firing rate modulations, and found that the reaches that had the subsequent highest firing rate were also the fastest reaches. And vice versa, those with the lowest firing rate and the slowest deceleration were the slowest reaches. So if you put the variation in peak velocity and the variation in deceleration together, what you end up with are stable endpoints. So given the early kinematic variability, it seems that a potential role for this variation in firing rates is to stabilize endpoints. So this is just a plot to show the gradedness of this code, where the highest firing rates are associated with the fastest deceleration and the highest peak velocities. Now, in a very simple model that just assumes a particular peak velocity where we can vary the deceleration, Matt went on to show that it's very important to vary the deceleration if endpoints are to be stabilized, because if you had an average unregulated decelerative phase of a reaching movement, but the early phase of the peak velocity or acceleration was variable, you would end up with wildly deviated endpoints. So this is shown here in these orange dots. If the peak velocity is low and the deceleration is average, you end up with undershooting reaches, and vice versa. If the peak velocity is very high and you have an average peak velocity, you end up with overshooting reaches. And so these data led to this idea that one of the feed-forward control sort of principles of the anterior interpose might be to regulate late phases of these reaching movements to enhance the movement precision. So that raises the next series of questions, which is really how is this computation made? Now, the most obvious target for asking about this question is just upstream of these neurons, the Purkinje cells, which form a major input to these cells. And so another wonderful student, Dylan Callum, who recently graduated, he focused his PhD studies on understanding how Purkinje neurons in a region of the cerebellar cortex that projects to the anterior interpose are active during reaching movements. And so he targeted his recordings to this interface between lobule 4-5 and lobule simplex that had been identified by Tom Otis's group as involved in forelimb control. And indeed, when he recorded Purkinje neurons in this region, he observed many neurons with simple spike activity that was modulated densely during the reaching movements. And so he went on to ask, straightforward questions about how might this activity be tuned relative to the kinematic parameters of reaches. And he performed a variety of lasso regressions and other kinds of regression methods and came up with this somewhat head-scratching set of observations. Basically, individual Purkinje neurons were, modestly tuned to individual kinematic parameters. And I would say that this was a bewildering set of observations because there was not any sort of clear tuning like what Matt observed in terms of velocity tuning. Now, I remember showing this to Reza when he visited, and Reza said very calmly, as he does, well, have you looked at this as a pooled population like we did in the oculomotor vermis? And I said, no, we haven't done that. That we don't, always have the, complex spike tuning, et cetera, et cetera. And he's like, well, have a look. And so I told Dylan, we should try and just look at these as a whole population. And Dylan basically said the exact same thing that I said to Reza, no, I don't want to do this. And I said, well, why don't you just do it? And so he, had noted for a long time that Purkinje neurons and putative Purkinje neurons, I should add. So not all of these cells are complex spike identified Purkinje neurons, but they all fall into similar simple spiking statistics. So caveat there. But among these Purkinje neurons and putative Purkinje neurons, there are cells that increase their firing rates during reaches and those that decrease their firing rates during reaches. Also seen in other regions of cerebellum during other types of behaviors. And when he mixed this entire population together and looked at the net activity rated by peak velocity, what he observed was the following. So here he has sorted reaches by their peak velocity that's shown in varying shades of purple. And what you can see is that the depth of the firing rate dip modulates with peak velocity. That is, the slowest reaches have the least net decrease in activity relative to baseline compared to the fastest reaches. So let me remind you that this is inverse to what was observed in the anterior interpost, okay? So first of all, there is a net decrease amongst the entire population, which is cool. And as inhibitory neurons that are pausing more or less, this would be a larger sort of reduction or suppression of simple spike firing rate associated with the highest firing rates in the anterior interposed nucleus, and vice versa. So this is the kind of thing that you see and you're like, wow, that's amazing. And so I can talk to you guys at the Gordon conference about this, this relationship more in depth. But for now, suffice to say that we think that this Purkinje neuron population could at least facilitate this kind of scaled activity in the anterior interposed. So now I'm going to talk a little bit about the timing of these different features of the reach relative to this pause. And here I'm using the term pause rather loosely. This is a suppression in firing rate. It's not a stoppage of firing rate by any means. But when is it happening relative to deceleration or peak velocity? That is it causing a change in peak velocity, or is it responding to peak velocity or early accelerative phases? So here, Dylan has plotted the time of the firing rate pause relative to a variety of features in the reach. So first of all, it happens after the reach has begun, and it happens approximately equivalently to the time of the max outward velocity. But importantly, it happens before the max outward deceleration. And so it is in a position to influence causally the decelerative phase of the reach. Now, one thing that we had observed in another study is that there is a relationship that is held between reach, outward velocity, and the decelerative phase of the movement. And so because there was kind of this then that relationship in the early kinematics and the late kinematics, as well as the timing of these firing rate modulations, it suggested that early kinematic variables like peak outward velocity might be contributing to generating these late phase adjustments. And this was interesting to us given classic observations by Hoare and colleagues of cerebellar patients, where they observed that the timing of agonist and antagonist contractions during very simple reaching movements was impaired in patients with Purkinje neurons. I'm sorry, in patients with cerebellar damage. They all have Purkinje neurons, presumably, but some of them might be dying. So in any event, this then that relationship of peak velocity and deceleration was reminiscent to us of classical conditioning, which of course is a form of learning that had been implicated as cerebellar dependent for years and years. So we're going to go on a little tangent here to talk about associative learning in the cerebellum, and then we're going to get back to reaching here. So when we're talking about classical conditioning, the classic paradigm that's used in cerebellar physiology, really developed by Dick Thompson, Mike Mock, and colleagues, is to pair a auditory stimulus. So this is a neutral, it's called the conditioning stimulus, such as a tone, with a noxious or reflex eliciting stimulus, in this case, an air puff. So this neutral stimulus coterminates with this unconditioned stimulus, it's termed. And after many, many pairings, the eyelid closure, which initially happens in response to the unconditioned stimulus, moves forward in time. It becomes anticipatory, such that the tone eventually cues that eyelid closure. Now, one of the cool features of this behavior that has been explored in computational models in very compelling ways is that the timing of this anticipatory eyelid closure does not happen here at the beginning of the tone. It happens late, right before the timing of the unconditioned stimulus. And so the question is, why does it happen there? How does the circuit learn to estimate this time interval to generate the eyelid closure at the right time. So Mike Mock and Dean Buonamano, as well as Javier Medina, developed this hypothesis that basically states that the granule cell layer, this is our prism, can act as like something like a prism in time. So the idea would be that it would take a neutral stimulus like a tone, which may be relatively invariant over time, and it would break that stimulus up into what is termed sometimes a temporal basis set. And the idea here is that each granule cell, which you could think of as a line here, is going to be responsible for communicating to the Purkinje neuron a little chunk of time, so that that tone is sort of represented in time across a population of these granule cells. Then, if there's a puff of air to the eye, the inferior olivary neurons fire. That, of course, induces plasticity events, which I'm just going to sort of loosely term LTD here, which are going to reduce the synaptic weight of neurons that were active only before the puff. Okay, so these guys out here are spared that plasticity event. They're going to be firing they're all going to be firing fine. Here, I'm going to conflate firing with synaptic weight. Bear with me here. In any event, so this sort of reduction in synaptic weight is going to be selectively applied to just the granule cells that were active right before the puff. So that happens, that reduces synaptic drive onto Purkinje neurons, as probably also gates inhibitory events there, but in any event causes a pause in Purkinje neuron activity, which of course is going to disinhibit the anterior interposed nucleus and cause an eyelid closure. Okay, so here we have tones associated with eye blink. Natural question, what does this have to do with reach adaptation? I could probably have put some other funny little symbols there. So what does it have to do with adaptation? So we hypothesize that the early kinematic variables of reach are essentially akin to a neutral condition stimulus, that this is potentially the predictor for a late phase adjustment. And so the working hypothesis goes like this. We have some kind of velocity profile for a reaching movement. It's represented in a temporal basis set in the granule cell layer. The limb overshoots. And so the inferior olivary neurons fire. And that causes an LTD event in the granule cell population. Those synaptic weights drop. The Purkinje neurons reduce their firing rate, anterior interposed activity fires, and now the next time this happens, there's a increase in that interposed activity, and there's a steeper deceleration. Okay, so that's kind of the working hypothesis. Now, how to test this hypothesis? So again, we're going to assume, and this is based on important data from Adam Hantman's group showing that M1 is essential for this behavior, that we're going to assume that these sort of movement initiation or signals emanating from M1 project down into the brainstem, including projections to the pontine nucleus. The pontine nucleus, of course, is the source of a massive number of mossy fibers, and so therefore is a good place to manipulate mossy fibers. And we're going to assume that the information coming into those mossy fibers is an important component of what the cerebellum learns to do like during reaches. The other thing is that if we manipulate these mossy fibers akin to manipulations of mossy fibers that encode the neutral condition stimulus in delay eyelid conditioning, we would predict that we can introduce sort of an artificial predictor into the reaching motor plan. So Dylan performed experiments where he introduced channel rhodopsin into the pontine nucleus and implanted an optical fiber in the region of the cerebellar cortex, again, that's involved in these reaching movements. And then he performed experiments somewhat similar to Matt in that he activated those fibers in closed loop with the reaching movements. Again, the Hampman group had shown that modulation of mossy fibers interfered with reach. But what we were curious about is whether this corrupted information could become sort of integrated into the motor plan in any way. So one of the key differences that Dylan introduced into his paradigm compared to the experiments that I told you about from Matt Becker was that instead of stimulating on a random 25% of reaches, Dylan stimulated these mossy fibers in a block. And the block trial structure is reminiscent of the kinds of adaptation paradigms that Reza has invented and have been used extensively in motor learning paradigms since. And so the basic idea here is that we have the mouse is allowed to reach uninterrupted for a baseline period, 20, 30 reaches. Then there's a stimulation block where we stimulate on every reach. And again, this is a brief perturbation of 50 milliseconds, and it's structured in a train. And then we enter a washout period. And so this is intended to just allow the expression of any sort of learned adaptation during that stimulation block. And so what do we see? So consistent with what was observed in Adam Hampman's group, stimulation of the mossy fibers during reaches caused perturbations in reach kinematics. In most animals, it caused an overshoot, but not in all animals. And some animals, it caused an undershoot. But in all animals, that initial perturbation went away over the block of stimulated trials. Then during the washout block, there was an undershoot in this animal relative to the overshoot that was initially caused by the stimulation. And then that undershoot eventually wore away as well. So this is a classic sort of arc of motor adaptation. And we observed this in a collection of animals over many, many sessions, suggesting that the cerebellar cortex may be able to adapt to errors that it is itself causing because of this corrupted information coming into the circuit. Now, there's another possibility, of course, which is that the rest of the brain doesn't want to hear about this garbage that the cerebellum is putting out, right? So we performed the same experiment, but using block stimulation of the cerebellar nuclei themselves. So here, the opsin is restricted to the excitatory output neurons projecting to the red nucleus. And so these are ongoing experiments done by a graduate student, Courtney Dobrat, in the lab. So she finds that the very pronounced undershoot that is induced by optogenetic stimulation of cerebellar output neurons is unchanged late in the block. And so with the same type of paradigm where there's absolutely very clear changes in the effective stimulation to the mossy fiber stimulation, there is not a change in that way to cerebellar output stimulation. So we do not think that the adaptation that we observe to mossy fiber stimulation is easily explained by the rest of the brain just tuning down cerebellar output. Okay, and it suggests that the site of learning may be in the cerebellar cortex, downstream of mossy fibers, but upstream of the output of the cerebellum itself. Okay, so that's enticing because it means that we might be able to observe this within session learning electrophysiologically. And so Dylan then combined experiments where he's stimulating mossy fibers and recording from Purkinje neurons and putative Purkinje neurons. And similar to the sort of split in neurons that respond, neurons that are increasingly active during reach versus suppressed during reach, he saw a similar breakdown of neurons that were excited by mossy fiber stimulation and those that were suppressed by mossy fiber stimulation. Interestingly, he also observed that this mossy fiber stimulation drove an increase in the probability of complex spiking. So this was exciting to us because it was reminiscent of what Shogo Ome had shown, which is that there may be this kind of relationship between what mossy fibers are doing and what climbing fibers eventually do. And we don't have much data to explore here, but one idea is that there is a sort of reduction in inhibition through the cerebellar nuclei caused by this from the nucleo-olivary neurons. And I'm not going to go into that, but our data are suggestive that there is a relationship there, as has been observed in monkeys. Okay, so tangent aside, then the question is, well, what happens during these adaptation blocks? So here again, Dylan is recording from Purkinje neurons and then stimulating these mossy fibers in closed loop with reach during a stimulation block. And here, because we have this sort of complex array of responsivity, we've broken these cells into groups, those that respond with a decrease in firing rate, those that respond with an increase in firing rate to stimulation. And we're going to watch what happens during this block of stimuli. So here's our sort of baseline average firing rate during baseline of these two populations. And then we observe what happens during the first 5 stimuli. So here we see that there's an increase in firing rate. Here there is a decrease in firing rate. And then if we track this over the next 5 reaches and the next the last 5 reaches of the block after that, we observe that there's this marching of the firing rate back towards baseline levels. And so you can see that in this pullout where we see the initial magnitude of firing rate excursion on the initial set of stimuli is greater than it is as the block marches on. So this suggests that the firing rates are re-normalizing back to those baseline periods. And you can see that here in this summary data, where here we're just plotting the net change in firing rate over the course of the firing rate block, the stimulus block. And you can see that there's a steady reduction in the amplitude of the firing rate excursion in response to stimulus during the block. So this is nice. This is sort of consistent with the idea that the Purkinje neurons may be key in expressing this within session plasticity. If we look at the total population then, so this is cells that are both responsive to the stimulation acutely and non-responsive to the stimulation acutely, and we look at that net firing rate, we find that that early stimulation is reflected in a opposite effect in the early washout period amongst the total population. And so again, this is consistent with this kind of rebalancing of the population activity across the population of Purkinje neurons. Okay, so what do we think is going on here? So I told you about the hypothesis of the temporal basis set that had been developed by Mike Mock and Dean Buonamano. This idea here that there's this temporal expansion of neural activity by the granule cell layer. So is this a reasonable hypothesis in the context of reaching movements? So my student, Jesse Gilmer, was really fascinated with this question, and he performed a computational study, examining how granule cells, which of course are very tiny neurons that have just 4 dendrites, might handle or process temporally varying mossy fiber inputs, those that would naturally occur during movements, which are themselves time varying. And so he reached out to, again, Adam Hampman and Britton Sauerbrey, who had performed recordings in the pontine nucleus during mouse reaching. The person who had done those experiments, Jeremiah Cohen, sort of gave us his blessing to use those data that they had previously collected. And so Jesse inputted those firing patterns into granule cells. And he found that there was an expansion of the temporal representation, or a smoothing of the temporal representation of those mossy fibers throughout the granule cell layer. He used a variety of other input statistics and sort of derived inputs to show that the granule cell layer readily stretches out or fills in time in response to time varying mossy fiber inputs, such that the temporal basis set is something of an emergent property that readily happens with very few assumptions in response to naturalistic input streams. And he later showed that this readily enhances learning of time varying signals. And so armed with this insight, Dylan, the other graduate student working on the Purkinje cells, built a computational model to explore what might be going on with learning in his paradigm. So here he's assuming reasonably a temporal basis set in the granule cell layer. So these are parallel fibers. Of course, the parallel fibers are the axons of the granule cells. So we can think of that as the same thing. And then he has sort of a balanced population of inhibitory neurons entering those Purkinje neurons. He then assumes that these granule cells are active at a small point in time during the entirety of the reach. They're not active during the entirety of the reach. They're active at a short amount of time. So then when we sort of pretend that we've got this this stimulation going. So this is our simulated stimulation. We struggled with this. Are we simulating the stimulation? So maybe there's a tongue twister in there somewhere. But the point is that that stimulation, of course, is also temporally circumscribed. It's only happening at a particular time in the reach. And that means that some of the neurons that are being activated during this stimulation are naturally active during this time. That's the part of the basis set when the stimulation is happening. But there are also neurons that are being activated during the stimulation block whose natural activation pattern is not at the same time. Okay, so this cell, number 1011, it's active at time 100 milliseconds normally, but it's being activated by the mossy fiber stimulation over here at time 250. And similarly, there are neurons that are being activated there are neurons that are active during the reach that are not being activated by the mossy fiber system. They're just active at that time. Okay, so we have three different scenarios at least. Neurons that are active at a time outside the stimulation block, neurons that are active inside the stimulation block and by mossy fibers, neurons that are active inside the stimulation block and not by those exogenous mossy fibers. So that's a mouthful, but bear with me. Okay, so then we ask this basic gradient descent model to learn to generate basically a small change in firing rate of the Purkinje neuron that's, loosely resembles this net pause that we observed. So under those conditions, when we activate the mossy fibers, we perturb the activity of those cells. But then over the course of the block, that perturbation is lessened, and we see opposite after effects. So then we ask, well, what's going on with the weight changes in this population of parallel fibers? And so not so surprisingly, we find that the parallel fibers that are stimulated by the mossy fibers, these ones are always associated with error, and their weights are turned down no matter where it is that they happen to be in the reach. But during the those that are active during the reach are very strongly turned down. Whereas those parallel fibers that are not stimulated by our mossy fiber stimulation pattern, they're unaffected. Their weights are normal until and unless they happen to be active endogenously during the perturbation, in which case their weights drop. And so that is sort of an important insight that we think is basically the driver of after effects. That is, these are the cells that are getting in the way. There's a kind of the unfortunate term that is maybe nevertheless useful to hear is this, they're kind of collateral damage in terms of plasticity being applied to them, even though they're not causing the error, they just happen to be active during the error. And so this model ends up recapitulating the magnitude of learning changes that we see during the block, as well as the after effects that we observe. So I'm going to end here to give us time for discussion. But the overarching set of ideas that we have explored here is that we start out with this problem in endpoint control that's seen with cerebellar damage. And we would argue that the naturalistic inputs that are going on in mossy fibers during movements that may emanate from motor cortex, as well as a constant stream of sensory feedback from the periphery, give rise to a temporal basis set in the granule cell layer, which allows for credit assignment associated with error to turn down synaptic weights such that Purkinje neurons can pause, or their firing rate can be suppressed around the time that errors might occur without a regulation of the activity in the anterior interposed nucleus that can causally scale deceleration, and ends up with this sort of tunable dial of limb deceleration in the output structure. So with that, I'm going to thank the many wonderful people I have the privilege to work with. Matt was really the initiator of many of these studies in his PhD work, focusing on the anterior interposed nucleus. Dylan Callum, sort of steady in the stead, worked on all the Purkinje data. Jesse also recently graduated and has a large paper in Journal of Neurophysiology on this idea of the temporal basis set and how it contributes to learning time series. Courtney Dobrat is doing work, of course, in the anterior interposed nucleus. And then I sadly didn't have time to talk about work from Abed Nashef, who is uncovering some incredible population level activity in Purkinje neurons. And so hopefully I'll be able to do that by the time the Gordon conference rolls around, and I look forward to that. Susan Coltman is doing incredible experiments in muscles that I also haven't had time to talk about, but she's recording motor units in active muscles, and they are a beauty to behold. And everyone else that I've had a wonderful time working with over the years. So thank you for your attention. I'll take questions. So.
Wonderful. Thank you so much. Let's open the floor and ask questions from Abby.
May I start?
Yeah.
I have. Thanks so much. That was fantastic. I guess one main hypothesis is that you're saying the climbing fiber will represent the error of overshooting and undershooting. Is this something you can observe? You should observe. Is there any relationship in the climbing fiber to anything particular that you can say, oh, it's an error?
Yes, yes, and no. So clearly the hypothesis predicts tuning to overshoots and undershoots. We don't necessarily see that, but we do see that the variance in the reach is much higher on trials with complex spikes. So there is a relationship, but it's not as directional as we might have assumed. Now, that could be because we're recording from mixed modules. It could be because there's a different kind of report that the climbing fibers are issuing. So I would say that at this point, we're a bit data limited in complex spike identified Purkinje neurons to make very confident claims about which of those possibilities we're looking at. But if you look at the bioRxiv update paper from Dylan, you'll see that this, that we do see that there's this very pronounced increase in variance of reaches that have complex spikes. So like I said, there's a relationship there, but it's a little bit confusing in the, in sort of the very crisp framework of directional tuning to them. Yeah.
Thank you for that. Rich has his hands up.
Thanks, Abby. Thanks for this open seminar. I feel bad I haven't taken advantage of it, but now the semester's over, I plan to be an attendee. So I want to go back to your first part of the talk where you laid out these two hypotheses, kind of the braking versus the directional signal.
Yeah.
And ask, could you really think of this as sort of a hybrid? Would be that it's a braking signal, but that braking signal is of the antagonist. And so if I stimulate on the outbound direction, right, I turn on the agonist with greater force, so I brake. But if I turn it on the return, now I basically am accelerating the agonist, so I get this reversal that you observe. And maybe that's the way you're interpreting it, but I thought that would sort of be a blending of those two hypotheses, basically.
That's right. And so what I would probably, the analogy, since we're using brake analogy, it would be like braking a car by throwing it into reverse. And so that's what that's essentially what I think. I'm a little bit more agnostic about whether it's a muscle tune, whether it's like specifically tuned to a given muscle. That I don't know about, but I absolutely agree with the principle that you're pointing out. So, right. But to me, it's different. Maybe the term brake, because you can brake a car by throwing it into reverse, is that it doesn't seem to be the case that the gain of the control signal is dropping.
Yeah, that's right. So it's not a generic brake, but it's a right. Brake applied in a specific situation, whether it's directional or muscle.
Absolutely. And so, and in fact, I use the vernacular brake all the time in the lab. So, so yes.
Thank you so much. Yoshiko has her hands up.
Hi, Abby. Thanks for the great talk. So I think my question is, like, how can I understand the oscillation by this gain control model?
How can you understand the oscillation? So the idea of the oscillation, and this is not mine, but it's just the sort of framework that's used in the field, is that if the sensory delay is very long relative to the movement, then so for example, visual feedback is slow relative to the output of the limb. Then the limb will tend to overshoot its target and then would have to overcorrect and then overshoot. And this is always because that sensory delay is too long. And so the idea here of the brake, thanks, Rich, is that by anticipating when to slow down rather than relying on the sensory feedback, the real-time sensory feedback, rather the system is learning to use the early sensory feedback, the early efference copy signals to anticipate what needs to happen, precludes it prevents the limb from overshooting because it's now no longer relying on that snapshot of sensory feedback that would tell it that it's at the target. Rather, it can move that sneak that back in time earlier so that it can anticipate its approach to the target. Does that answer your question?
Yeah, so that oscillation is kind of like several movement. It's not like single movement result. Like move the first one and it overshoot another movement to get the target, and another movement and another movement, another movement. So
Yes.
it's a kind of series of movement, not like single movement and oscillate.
I don't know, maybe I'm missing a nuance there, but yes, you're right that it's a series of, I would say that those movements to touch your nose, there's a single goal, right? Which is to, just touch your nose. And yet the oscillation around that endpoint, you could decompose it into multiple sub-movements there. But yeah, does that make sense?
Yeah.
Yeah.
Thank you.
Thank you.
Shogo, you have a question?
Thank you, Lisa. Thank you very much for your wonderful talk, Abby. I really enjoyed your talk. My question is a simple question. So, is there any other neurons controlling other directions in the cerebellum? So, because you are related to Rich's question, the population is very specific in the it should work breaking in a very specific situation, but I think we need other directions.
Yes, I agree with you. And so this has been the most asked question, and it's frustrating that I still don't have an answer for anybody. No one will take the bait on this project. I'm like, oh, we should do this. Nobody wants to do this. But let me just spell out a couple of options. So I think the most likely scenario is that there may exist an accelerative module that's for the limb. I mean, I think that all these effectors are going to have these modules. That there may exist an accelerative module for the limb that is outside the anterior interposed, because we don't see any evidence of, salt and pepper, or, that it matters, what the amplitude of the stimulation is, like that we would bias to outward or inward. It's always this inward pull. So we are in a spot where this is the very, very robust and clear result. So if there is an accelerative module, we think it's really somewhere distinct. A. B, the nuance to that idea is that there's something interesting in the bidirectionality of the relationship of anterior interposed to the reach velocity. And it seems somewhat distinct from what, the framework of delay eyelid conditioning, where I don't think that there's a recognized role for inhibition of those cells in that behavior. I mean, I don't know if the eyelid is getting like higher and higher. We're all dealing with these biomechanical constraints, but nevertheless, the possibility then remains that maybe there's something about that bidirectional control that could be exploited for the accelerative modulation, right? So I don't know. I mean, I don't know. I think there must be an accelerative module, but.
Thank you so much.
Thank you so much.
Thank you, everyone. I guess if it's okay, let me ask the final question, because we should end. Abby, can you teach us a little bit about what we know regarding the physiology of the temporal basis set that you described? Do we know that there is this transformation that takes a muscle fiber input and makes a temporal basis set?
We don't know it very well, but there are data that are suggestive that this idea is probably on the right track. So imaging experiments that have been done by Andre Giovannucci and Mark Wagner, electrophysiological recordings that have been done by Paul Chatterton and Angus Silver to some extent, and Mike Heuser. They are all pointing in the direction that there is a time varying signal in populations of granule cells, and that granule cells fire in brief bursts of activity during sensory stimuli. And so I would say that the sort of slam dunk electrophysiological population recording that shows this transformation doesn't exist yet. But I think that there's a number of pieces of empirical data that are pretty warm on that direction. So I don't think it's going to be wildly incorrect. I think there's good reasons to think that that's approximately right.
Thank you so much. Thank you, everyone, for joining us. Abby, we're really grateful for this.
Thank you so much, Reza, for doing this. I want to just echo Rich's comment that this is a real gift for the community, and so it's fun.
Well, you are the gift. Have a wonderful Tuesday.
Yeah. Have a wonderful December.